Good morning, everyone. Good afternoon, good evening. It depends on where you are. I'm Professor Tianhong Cui from University of Minnesota. It's my great pleasure to chair this biosensor section of the Design of Medical Device Conference. As you know, this is the 19th Medical Device Conference this year. We'll move it online because of the coronavirus this year. So in this section, we will have three speakers from Mayo Clinic, University of California, Davis, and the Chinese Academy of Sciences. Each speakers will have 20 minutes for presentation plus a couple of minutes for questions. So for all your questions, you can type in the Q&A section. So I will help you to read the questions from your audience. And then the speaker will answer the question accordingly. So the first speaker is, let me introduce the first speaker, Professor Alex Reversion. So today, unfortunately, he cannot show up here because he has a panel review at a governmental funding agency. So he pre-recorded his talk. So let me introduce Professor Reversion from Mayo Clinic. So he is a professor in biomedical engineering and physiology at Mayo. And he has been working in the field of microsensors, microfluidics for the last couple of decades. He was actually a professor at the University of California, Davis for years, go all the way from assistant professor to full professor. Then he moved to National Science Foundation a couple of years as a program director in the biosensor division. And then after his service at the Mayo Clinic, he moved to Mayo Clinic at Rochester, Minnesota. Ever since, he has been working on a lot of work, biosensors, microfluidics for cancer diagnostics and treatment. So today, his talk is pretty much focusing on cancer treatment. So I will ask Gary to turn on his video. Gary, please.
Hello. My name is Alex Reversion. I'm a professor of physiology and biomedical engineering at Mayo Clinic, and it's my pleasure to have a chance to speak to you about some of the work that my lab is doing in developing organoid cultures for personalizing cancer treatment. I apologize I'm not able to present this live. I have prior engagements at the time of the Biosensors session, but I appreciate the fact to be able to speak to you in this pre-recorded seminar about some of the work that my lab does. So let me go into presentation mode here, and I'm going to turn off my video for the duration of the seminar. Just a second, I'm turning off my video. I did it. Okay, great. So now let me tell you about reason why we are developing microfluidic cancer organoid cultures. I'm forwarding slides. There's a slide delay here. Delay. Here we go. So the motivation for this is that oncologists increasingly need ways to personalize chemotherapy administration. So there's a need to model how a given patient is going to respond to chemotherapy or molecularly targeted therapy. There are tools that are increasingly available to oncologists. For example, sequencing is something that is frequently done on patient tissue where RNA sequencing is used to analyze presence of genes that are upregulated or aberrantly expressed, and based on this, on presence of these genes, one could come up with therapies. However, despite the fact that sequencing narrows down the therapies that could be used on a given patient, it does not give a single one consensus therapy. There are always several options that oncologist is faced with. So even with sequencing, even with this new new way for precision medicine, even with sequencing, one has to still have a way to test therapies and screen therapies. And so there is a need to develop personalized models or avatars for predicting patient-specific drug responses. And one model that is very popular and very commonly used is called patient-derived xenograft, or PDX. And in the case of PDX models, you have patient tumors or cancer cells that are engrafted into mice and propagated, expanded in mice. And this is an excellent model. It's very popular, but there are challenges and disadvantages to this model. The process of engraftment is oftentimes lengthy. Oftentimes it takes 6 months for patient cancer cells to engraft in a mouse, and then you can imagine that each mouse is an experimental condition. Each mouse would represent a concentration of chemotherapy or a different drug that would be tested. So it's one mouse per experimental condition. And if you have multiple experimental conditions, this makes mouse models expensive, and it's difficult to test different conditions. I work with oncologists who are treating aggressive metastatic cancers where they do not have time to wait 6 months for tumor to engraft. They would like to know much more rapidly how a given patient is going to respond to chemotherapy or molecularly targeted therapy. So spurred by this need for more rapid turnaround in terms of being able to test therapies, anti-cancer therapies, the field of cancer research has come up with this concept of cancer organoid cultures. So these are in vitro cultures. These are organoids, which represent sort of three-dimensional constructs with dimensions on the order of hundreds of micrometers that are grown in dishes, and they are grown using protocols that are adapted from the field of developmental biology and from stem cell cultivation. And presumably what happens in this organoids is you have a cancer stem cell compartment that is propagated, and you have the structures that are growing in the dish. So throughout my presentation, I would go back and forth between organoids and spheroids. In my mind, they are synonymous. Some people are more sort of purist about this, and for some people, organoids represent sort of stem cell containing cultures, whereas spheroids are more monolithic and more differentiated cultures. For me, I go back and forth, since spheroids, organoids are similar in my lexicon at least. But the thing about organoids is that they are typically grown in standard multi-well plates, 6-well plates or 12-well plates, and the standard cultures utilize large volumes, and the standard cultures require large numbers of cells to establish organoids. And so what the challenge that we are facing is that there is tumor tissue that's readily available to us. So oncologists and radiologists frequently, routinely collect needle biopsies from patients, and these needle biopsies contain needle cores, which are minute sort of a sliver of tissue, as shown here, perhaps few millimeters long and less than a millimeter in diameter. So this needle cores come from 16 gauge or 18 gauge needles, and they are collected routinely. They are available. They are collected prior to chemotherapy. and they are typically collected for pathology testing, and they can be made available for organoid cultures. But this amount of tissue constitutes perhaps 100,000 cells, and it's really not enough tissue to establish standard organoid culture. So it's really difficult. It's really wonderful that there is this source of patient-specific tissue that's available, but it's really difficult to establish standard organoid cultures using this patient tissue just because there isn't a lot of it. So what we are thinking about is, instead of standard multi-well plates to form organoids, we would like to use microfluidic devices. Microfluidic devices are appreciated as tools that allow one to do more with less, to do more with fewer cells, and to do more with smaller volumes. So what we are doing is we are creating this micro-well arrays inside microfluidic devices. We functionalize this micro-well arrays with this pluronic and non-ionic surfactant that minimizes cell attachment to the surface, and cells end up being forced to attach to each other and form spheroids of the type shown here. And these are images of spheroids forming inside microfluidic devices, inside microwells. We have single cells here at day 0, and by day 3 we have compact spheroids that are forming. And so the idea here is that we have needle cores that are made available to us by our oncology and radiology friends, and we would like to be able to digest these needle cores, to form them into spheroids or organoids inside microfluidic devices, and then test multiple drugs based on an input of a low number of cells, say 100,000 cells. So what we are doing to achieve this is we are creating microfluidic devices that contain multiple cell culture compartments. So there are 8 cell culture compartments that appear in blue in this device. Each cell culture compartment has 10 wells for forming spheroids. Wells are about 300 micrometers in diameter, 300 micrometers in depth, and our spheroids or organoids are on the order of 150 micrometers in diameter. So these devices contain multiple compartments. They also contain microvalves, automated microvalves, and these microvalves can be reconfigured, where the device can operate in two configurations. Serial perfusion, where we perfuse all chambers, and this is used to seed cells. So we could take a small number of cells and we could seed chambers in sequence to minimize loss of cells. And then we can reconfigure the device. We can sequester compartments. We can separate compartments from each other, and we can perfuse individual compartments with different solution, with different drug or different concentration of drug, of the same drug. So we have this reconfigurable microfluidic devices that hypothetically should allow us to do more with a small number of cells available in a needle core. So here is a video showing operation of the device. We are operating in perfusion mode here, in seeding mode. So we seeded, we flushed all 8 chambers with, say, the cell suspension, and now we are going to reconfigure the device. We are going to reconfigure the device. We actuated micro valves and compartments. These compartments are now separate, and these compartments can now be addressed with different drugs. In this case, different food dyes for proof of concept purposes. So one question that we asked was, is it really feasible to seed this 8 microfluidic compartments using an input of 100,000 cells? And once again, 100,000 cells is important because that's approximate number of cells available to us in a single needle core from an 18-gauge needle. So here is an experiment that we did. In this experiment, we used GFP-expressing breast cancer cells, and we seeded, or we infused 100,000 cells into our device versus 25,000 cells. And then we characterized diameter of spheroids forming in chamber 1, closest to the input of cell, versus chamber 8, farthest from the input of cells. And so here we see that for 100,000 cells, we have uniform spheroid formation from chamber number 1, that is closest to the input of cells, to chamber number 8, which is farthest from where the cells enter into a microfluidic device. So indeed, with a small number of cells, with 100,000 cells, we are able to seed 8 chambers. The next question is, okay, how are spheroids performing in our microfluidic device? Are they doing well? One way to answer this question is shown here. We are looking at morphology of spheroids, and we see that at day 7 and at day 14 we have compact spheroids in microfluidic devices. And when we do live dead staining, we see that spheroids at day 14 are alive. They fluorescent green, and there is very little dead cells, as indicated by minimal red fluorescence in the spheroids. So we also, in addition to looking at morphology of spheroids, we also went a little bit deeper and characterized some of the epithelial cancer markers that should be expressed in cancer spheroids that are maintaining phenotype. And here we show staining for AbChem, an epithelial surface marker, and cytokeratin, an intracellular protein, a cytoskeletal protein, which are both indicator of epithelial cancer cells. And we see that both of these proteins are well expressed in our spheroids, suggesting that phenotype is maintained in microfluidic devices, and this is after 7 days in culture. We also see staining for Ki67, which indicates proliferation of cells. So our spheroids are forming well, they are maintaining compact morphology, B, they are expressing correct cancer markers at day 7 in culture. So in addition to this characterization of epithelial markers, I wanted to show you what spheroids look like in matrigel cultures versus in microfluidic devices. So this this column here are 3 different tumor tissues cultured on matrigel. And you can see that this construct, this cellular organizations are irregularly shaped and irregularly placed on in standard matrigel coated dishes. Here you actually see that there is no spheroids forming at all. So in this case, you do not have spheroid formation. Whereas in our microfluidic device, we have regularly shaped and regularly placed spheroids. And in this case, where you do not have spheroid formation on matrigel, you do have spheroid formation in our microfluidic devices. The reason why spheroids are important is it's relatively easy to monitor changes in geometry, expansion or shrinkage of spheroids during growth or shrinkage when they are challenged with chemotherapy. So being able to look at defined geometry of cancer cells has benefit because it makes it easier to monitor responses to chemotherapy by microscopy, via microscopy. So in summary, we have been working closely with oncologists at Mayo Clinic, and we are part of several IRB protocols, and we have demonstrated ability to take tissue available from needle biopsies and to form spheroids or organoids from this tissue, both on matrigel and in microfluidic devices. And we show that oftentimes spheroid formation happens better in microfluidic devices, and spheroids do better in microfluidic devices compared to gold standard matrigel cultures. You may ask, how do we carry out drug testing in microfluidic devices? This is one example where we have spheroids that are exposed to a molecularly targeted therapy called vemurafenib. And what we do here is we do live dead staining, and we characterize or we quantify a number of live cells fluorescing in green versus number of dead cells fluorescing in red as a function of chemotherapy concentration. So there is more dead cells for higher concentration of vemurafenib. Similarly, these are lung cancer organoids in microfluidic devices, and this is a different drug, trametinib, also a molecularly targeted therapy, and you can see that there is a higher number of dead cells in the presence of higher concentration of this drug. And so we can quantify different concentration, or we can quantify how different concentrations of this drug affect cytotoxicity, affect viability of cells. So to briefly summarize first part of the talk, I demonstrated to you that we can form and maintain primary cancer spheroids in microfluidic devices. We achieve phenotype maintenance for at least 14 days for most biopsy samples. And this phenotype in our devices is similar or better to gold standard matrigel cultures. We are developing well-enabled microfluidic devices for testing several drug concentrations and combinations. And what we are really keen on doing is addressing a question of whether organoid cultures can be predictive of patient drug response. And that's one of the reasons for using our microfluidic devices, that conceivably we can test anti-cancer therapies within a short period of time, such that our oncology friends would be able to take a therapy predicted by our cultures and administer this therapy to patients. So these are the types of studies, these are the types of clinical trials that we are discussing with our oncology collaborators at Mayo Clinic. So, so one question that's a bit better aligned, that's a bit more on the side of biosensor development, is how to analyze function of cells in microfluidic devices. Individual cell culture compartments that I was showing in this 8-compartment microfluidic device, individual cell culture compartments are probably on the order of 1 microliter or so. So how to analyze cell function where cells are secreting signals into 1 microliter of media. My lab has a long history of developing sort of de novo biosensors, electrochemical biosensors, sometimes optical biosensors, but each biosensor takes several months to develop. It's kind of challenging. On top of that, what's kind of exciting for me is that there is a plethora of bioassays that are available, biochemical assays that are commercially available. So these assays come from Sigma-Aldrich or Thermo Fisher. They are robust, they are well characterized. The problem is this commercial biochemical assays that are off-the-shelf assays are designed for large volumes. So one cannot use commercial assays, commercial biochemical assays, to analyze sub-microliter volumes. So that's kind of a challenge. And so what I would like to show you is how we are thinking about taking off-the-shelf biochemical assays and miniaturizing them to make them suitable for analysis of cell function in this microfluidic devices that we're developing. And so this is sort of a video, a little bit faster than real life, perhaps, video that shows an individual who is working on this conventional mix and read assay. And essentially, the point here is you have many assays, enzymatic assays, for example, where you take a sample, you mix this reagent, and then a few minutes later you have development of color, either it's a visible color or fluorescence. And so these assays are robust, but they are labor intensive, and most importantly for us, they are really designed to work in tens to hundreds of microliters of sample, whereas we have sub-microliter samples in our microfluidic devices. So to address this problem, to address the need to miniaturize liquid handling for mix and read assays, we developed a microfluidic device, for mixing and in-droplet analysis of bioassays. So essentially what we have here is we have a co-flow of two aqueous streams, one containing assay components, reagents for the assay, and another containing sample. And this aqueous streams are injected into an oil phase here, and they form droplets. And so what we have is we have an automated microfluidic device where we could load different reagents into different sets of droplets, and where droplets are contain wells where biochemical reactions are happening. And so now we have an assay, we have a system that allows us to analyze, to run multiple biochemical assays on nanoliter volumes, right? So now this system is appropriate for the volumes that are available in our microfluidic devices. This is kind of a demonstration of device operation. We have several inlets. These inlets are each controlled by individual microvalves, individual addressable microvalves. Inlets are loaded with reagents. We also have a sample stream, and so now what we do is we actuate valves in pairs to allow for two streams to flow and to eject these streams into droplets. So here we have a blue stream and yellow stream flowing together, and now we have a red stream and a yellow stream flowing together. And yellow stream is our sample. This is our sample input, and this is our reagent input, and this is our reagent input as well. Okay, and so we can switch which two streams are meeting and mixing and being ejected and being formed into droplets. So as I mentioned, there are lots of assays that are commercially available. One example is an LDH assay, which is an enzymatic assay to quantify concentration of lactate dehydrogenase, which is an enzyme that reaches out of dying cells, and this enzyme can be, or the concentration of this enzyme can be characterized via this multi-step enzymatic reaction that results in appearance of fluorescent substrate. Okay, so one can create calibration curves of concentration of cells versus fluorescent signal, and this is some of the examples of in-droplet signals from different concentrations of cells that were analyzed off chip. So now what we can think about, so one application that we can think about is taking a cell culture system that contains, say, our organoid cultures, and transferring volume, a small volume from this cell culture system into a microfluidic device for analyzing, for in-droplet analysis. So that's one thing that we can do, and we have done it. However, the problem is in the process of transferring from this cell culture platform into this bioanalysis module. In the process of transferring liquid, we end up diluting sample by an order of magnitude or more. So what we wanted to do is we wanted to integrate in one device a cell culture platform where we are forming organoids and a droplet generating module where we could sample media conditioned by these organoids and analyze this media for presence of indicators of cell function or cell death. And this is a device that we are developing, this kind of work in progress. Cells are cultured here, or organoids are formed here, and the bioanalysis module is here. And so what we are showing in this graph is that when we use a fluorescent reporter, we have a stable fluorescence signal for hundreds of droplets when we collect media from this integrated device. Whereas if one were to have two devices connected together, so not a monolith device, but two devices connected via tubing, A, signals are a lot lower, and B, the signals are decaying precipitously. So with an integrated platform that we are developing, we can sample minute quantities of cell conditioned media, sub nanoliter volumes, and not dilute the concentration of signal in the sub nanoliter volumes. So second summary or summary of second part of my talk is I demonstrated to you that microfluidic automation can be used to analyze small volumes of cell conditioned media. Multiple assays with built-in repeats, positive and negative controls can all be run on sub-microliter volumes. Platform, this is a platform technology. It's a liquid handling technology as opposed to a biosensor per se, meaning that it can be used with commercial mix and read assays. Fluorescence and absorbance works well. Finally, I would like to acknowledge the people who did the work. Neda is a talented visiting graduate student in my lab. Gulnaz is a project scientist in my lab. They're both working on the biological aspects of cancer spheroid formation characterization. And Alan Gonzalez Suarez is a bioengineer who develops automated microfluidic devices in my lab. And so with that, I would like to acknowledge the funding from NIH, State of Minnesota, and Mayo Clinic. I would be happy to entertain questions about questions about this presentation or ideas for collaboration, and you're welcome to reach out to me at this email address, and I would be happy to answer any questions that you may have via email. Thank you very much. It was a pleasure to speak to you.
Okay, let's thanks Professor Revizin for his interesting talk. Actually, he used up all his Q&A time. So if you have questions, of course, you can directly send an email to Alex, or you can send a question to me, and I can forward your question to Alex Revizin. Let me introduce the second speaker, Professor Ting-Rui Pan. Professor Ting-Rui Pan is he's working for biomedical engineering at University of California, Davis. And he got his PhD from University of Minnesota. And he moved to California. He has been working on micro systems, microfluidics, and he did a lot of work in between academia and the industry. And he is elected fellow of American Institute for Medical and Biomedical Engineering. He also a fellow of Royal Academy of Chemistry. And with this, I'd like to give the podium to Professor Pan for his talk. So Ting-Rui, please.
Okay, thanks Tianhong for a very kind introduction. It's for like to attend I attended the very first one when I was a graduate student in Minnesota, and no Tianhong there. And then I was attended Tianhong's panel a couple years back as well. And what a coincidence is, Dr. former speaker was my former colleagues. We worked together on biosensor and microfluidics, which are quite nice. So he actually helped me with some of the introduction. I probably can save my time to give you the more focused talk on the things we're doing. So, just, connected to, what the world with the biosensor, microfluidic has been, important buzzword today. And, we want to see, at our point, what we are interested to see is how the microfluidic has been developed and how is the technology will be eventually, come to the users. Microfluidic technology is being, known for about 40 years now, since the first, microfluidic sensor has been invented on silicon substrate. And with this being long history, microfluidic meet with one problem, it's a buzzword in technology, but it hasn't been fully utilized in many, research laboratory. Although some, commercial succeed, but it's still quite expensive and difficult to be utilized. What we promised for the past 40 years for microfluidic system to be used is one incentive is the system automation, and secondly is attached to the micro. The volume can be very, very small, and the total system can be quite, integrated and miniaturized. More, come from the cost side, the reagent will be consumed less, and we can take advantage of certain, micro and nano scale phenomena like faster diffusion, greater aspect ratio of the structures can, promote more reactions. And eventually, because of shorter distance, it can, reduce the reaction time. But all these are great promises, but we still need problems, right? So what's the problem? If we look back through the history, the original, analysis was done in a way, we can use a titration system, which has been, there about 200 years. And lately, the operation biological lab is since 1960s, and where we use a standard pipette system, which pipette actually is making the conventional titration system into a mobile and a fashion, and can do different volumes in a small range. And lately, one of the striking point is we actually come up with a different format of the reaction. It's using Droplet. So the Droplet microfluidics has been, advanced and populated in the field of microfluidics for many years, and including, the former presentation with a lot of microfluidic droplets to be reactor force. So there are a couple formats of the Droplet microfluidics. Right now, the digital is also called a digital because it's compared to the continuous flow. This Droplet reactor more like individual reactors, and the reaction fashion can be digitalized. So it becomes a mainstream of microfluidic reaction and can do a lot of things. The origin of the digital Droplet microfluidics was started with this very, very interesting phenomenon. It's called electroweighting on dielectric. So basically, when we apply a voltage, the droplet shape can be changed, and it can be driven to moving directions. It can be splitted. It can used to be into a mixing reaction, and it can do a lot of operations on digital. And there are actually a few companies that work on technology, including Illumina and BGI. They already have some products based on the eVolt. Another very popular technology from the digital droplet direction is using the flow focusing. Okay, the T-junction technology. Bio-Rad is one of the pioneer to use this to do their digital PCR system. And recently they just introduced their system of the PCR. Besides these two mainstream technologies, there are also technology actually people are using the microfluidics to do a drop on demand. So drop on demand means instead of generating continuous flow or use electrode to control the flow. So basically they can print things and into a droplet. So this actually represents the kind of inkjet development even before microfluidics. Another recent trend is people actually use other energy source like acoustic powering. So they can actually turn into a droplet flow, through a non-contact mode. So the acoustic wave, like the surface acoustic wave and bulk acoustic wave can all be utilized to generate a string of the droplet with the same size. So lab site in Bay Area is, release a machine can shooting a tiny droplet from 2.5 nanoliter to 25 nanoliter into a very small droplet string from one well to another well. So they can actually doing blended things based on this technology. We actually came up with an idea a while ago, and one of the intention is actually all the system you have seen before, it's quite expensive and complicated. Can we do something really simple? Can we do something, you can operate in the laboratory and, manipulate the fluid with nanoliter precision? So we came up with the idea where we actually utilize old-fashioned dot matrix printer, which actually can deposit nanoliter droplets, but with all the multiplexing you can utilize in a dot matrix printer head. So on dot matrix printer head, typically you have a bunch of the actual electromagnetic actuator, which can move up and down and squeeze a ribbon. Typically it's a color ribbon to deposit a color on the paper substrate. In this time, and we turn the paper cartridge into a microfluidic cartridge. And inside we loaded different chemicals, and each striking of individual channels will leave a trace on the substrate with a droplet. So using that method, we turn our existing, the dot matrix printer uses existing software. We can actually generate a microfluidic printer. So this was the original microfluidic printer looks like. And it actually was a very, very simple construct. And you can see there are 5 channels, and each loaded in with different chemicals. And on the end of the channel, you can see there's a tiny nozzles there. And when you load the liquid into the channel, and there will be positioned dot matrix printer head and the position to the tip, and each of them will be able to control by the computer and strike a droplet to form different images. And we use the motion stage and the control system of the microfluidic printer and to generate any combination of the chemicals and the formation of the chemical patterns as shown there. So to compare the 4 technology we mentioned about up now is the electroweighting based technology. Highly customizable, but the cost is very high. Flow focusing is pretty easy to make. The throughput is high. They can generate thousands or 100,000 droplets in a shorter period of time. But to control the whole system and make a customization could be very difficult with the consistency you like to have. Dot matrix has been very popular with addressability, but the cost of making the system and the control of the system is quite difficult. The acoustic ejection, of course, the cost is extremely high. The typical system on the market, they pay $250,000 to acquire, and it's very difficult to customize. And if you look at the control system, it's readily available. You can actually buy from the off-the-shelf. And the microfluidic chip you have to make is a pretty standard. You don't have any interconnections and other things. Can be actually quite adaptive to different systems. So given the system we have developed, so we're looking at what can we do. And one thing we're thinking is, can we target a conventional application which requires higher precision and a non-contact mode of the printing we can deliver. So the target first is the digital pipetting. So instead of actually using the conventional principle of the pipetting, pipetting actually has meted a problem. The conventional pipetting utilizes the positive displacement method, which actually just use the air chamber to withdraw the liquid and dispense the liquid. It works well with, like a 10 microliter or above volumes because the air volume doesn't really matter. Surface tension is not a problem, but it does have the issue with the inaccuracy under 1 microliter or around 1 microliter. So the reported error for this type of operation is, between 20 to 50%. So any on the market, the pipette system, you can see they will claim, at about 1 microliter range, the error could be up to 50%. So 50% is really, you consider it's probably not very accurate. There are a lot of influence factors like temperature, the surface tension on the tips, and the residues, and how experienced you're doing this practically. One actual solution could be solving this is how about we use Droplet. So Droplet solve this problem in a couple folds. One is Droplet can achieve much smaller volume, okay? Much smaller volume actually can give you the precision directly. But also, if the volume is much smaller, the total errors of the individual volume will be divided by, the number of the droplet of the square root, so the statistical mean. So actually you can, have a smaller variation just because the droplet is smaller and you have a number of droplets. And 3 is actually the droplet printing is actually non-contact. So that directly avoids the problem of the residues. So, and also it prevents any crosstalk or any contamination issues we might meet in the conventional system. So one of the typical experiments that you actually do is actually in the titration, you have to generate the serial dilution, right? So, I mean, that's a very typical operation in a chemical lab. So you have to make the concentration, half the concentration, quarter of concentration, 1/8 of the concentration, continuous with multifold to create probably some of the response curve as a standard curve. But this actually can be beautifully addressed by the droplet printing because now we turn everything into digital. Instead of doing serial dilution, which actually you are doing that 1 by 1, half, we can actually directly calculate the desired concentration and to know the minimum, resolution of the digital printing. So instead of doing, 1 out of 100 by diluting, 8 times more, so we actually can do 1 droplet versus 99 droplets. So we can do a combination, and then we can actually accurately control the dilutions level. And more, this we can give any random, or desired concentration by just numerically combining the buffers and the targeted dilutant. So to do this, we actually first think about the user experience because most of the people in biological lab, they are not experiencing anything besides the pipette. That's another reason, microfluidic is very difficult to be utilized in a conventional biological lab because people requires a lot of, technology learning curve to catch up to how to use microfluidics. So that's why microfluidics are typically restricted to certain labs and not to many of the biological labs. So here we actually making a compatible system to the existing pipette, and then by adding a jacket to them. So the jacket actually is the green part you can see here. And so we actually make a conventional pipette tip connected to a microfluidic pipette tip. At the same time, we actually have the electromagnetic actuator to deliver the droplet into a very, very small format. So what you do is typically you have connected your microfluidic pipette jacket and install that in a pipette. The program has been downloaded previously. And then you actually can use the pipette system to restore the liquid. And afterwards, you use the microfluidic printing system to deliver the chemical concentrations. And the A shows you how this microfluidic pipette system works. So you can restore the liquid from the channel 1, and then actually you will deposit a very small droplet from channel 2. So the next one give you a video show how this works. So this is actually the microfluidic jacket that you will use. And with the jacket, you will put the microfluidic the control program downloaded into the chip allocated on the top of the program. And then later you can put the microfluidic pipette on top of it and install the pipette there. So with the pipette, you actually can restore the liquid through the tips. Remember, the tip has 2 parts. One is the conventional tip, which actually restore the liquid, and then there's the electromagnetic actuated part, which will allow you to deposit the droplet through the orifice there. So this actually dispensing, actually, when you're doing the dispensing, you just need to push the button, and then the pre-programmed, allocation will allow you to do that. So in the next generation, we actually, make the system even more simpler, and integrated the pipette, into one set of the system. So where we have the actuator on the side, and then we make one pipette system into a tube shape directly, instead of have two systems to work, channel 1 and channel 2. Now we actually can have only one channel with sort of liquid, and dispense from the same channel into the droplet stream. The only problem you have to address here is to make the tip very hydrophobic, because if the tip is not hydrophobic enough, it will not allow it will still have some residues on the tip, actually will affect your performance of the droplet deposition. And here we use a laser fabrication to make the super hydrophobic nozzle head, and where we have micro patterns and nano patterns, both are very highly hydrophobic. And so you can see in the edge, this is a super hydrophobically treated the nozzle tip, and the other one is not. So you can see clearly hang over of the droplet around the tip head, which will actually influence your deposition performance. So this is a high-speed, droplet deposition image. You can see the droplet are forming a very spherical shapes, and with a highly, regulated the volume distribution with a very small CV. The CV is always smaller than 1%. So that means the individual droplet has less than 1% CV. So if you have aggregated volume with multi-drop deposition, you actually can achieve even smaller, as the large number of the droplet actually will average out each arrows generated from the droplet system we generate. So from the picture D, you can directly see when you use a conventional pipette system, which actually is drawn in blue, and they have quite large arrows from the random arrow, and also quite appreciable system arrows, which actually is coming with the pipette system, particularly dealing with small volumes less than 1 microliter. But when actually you use the droplet system, actually you can see in the smaller volumes, the systematic arrow can be very, very small. So we can keep actually a very small systematic arrow, and eventually push to the limit of the droplet printing system to a smaller limit. And this is when actually we are dealing with the serial dilution, which actually in the conventional way you do 1 by 1. And you can see the variation also can quite significant, and particularly in a very small concentration volume. But when you actually do a digital pipetted format, actually you can achieve a very, very, very small variation in the digital format, which actually is mostly caused by the satellite droplet and a certain spills from the droplet during the printing process, but which has already shown you a very consistent performance over different volume of the concentration. We also tested the drug concentration in a typical drug-resistant array, and where you can actually test the cell viability, use the different drug concentrations, and so the smaller volume of the digital pipetting system, we can create a much finer volume control in a low concentration profile, which actually use the conventional pipette system. You're only limited to 1 micromolar concentration range. So this actually allows us to do a very small volume deposition and highly programmable and easy to use, and it's very compatible with the current pipette system people have, and reduced to the human error with pre-programming. The nice thing about this is eventually this is digitalized. We can actually extend the digital microfluidic one more step. So what's one more step? So what we have seen in the last video is we can see the people are only using the system into manual system. Basically, you still need to have a person to operate. But the person operation is, as you have seen in the video, is just push the button and move around. So this can be well, connected to a robotic system where the microfluidic operation can be completely automated. So then we created the next generation, which we call the microfluidic Cap-to-Dispense system, which we can connect the containers of liquid storage to a micro robot. The robot actually can handle everything from the dispensing, mixing, and everything into one actions. So where we can actually use the user to remote control the whole operation of the lab operation. So the principle is now, how can we make the microfluidic on a robot? So what does the robot require to have to operate the liquid? So we actually look at the robotic arms. So this is a typical robotic arm, and we add in the pneumatic system to operate a typical F&F tube. And inside the tube, there are liquid contained, and then we are turning the continuous medium into a droplet. So in this system, we actually implement two controllers. So one is on the left hand, one is left finger, one is on the right finger. So one on the left finger is actually provide the higher pressure with a pulses flow, so which will allow you to generate a droplet in a short period of time. On the right finger, so you have lower pressure control, which actually is pushing the liquid to be loaded on the tip of the nozzle. So here's the pneumatic circuits. You can see when you're actually pushing the flow, the low drive on the right-hand side is pushing the flow towards the tip. And when you turn on the high pulse, the pulse gives you a pulsatile flow and will eject the simple droplet from the nozzle side and into the droplet format. So with that process, we are adding the computer vision to the system, so the actually robot have an eye to see what chemical it is. And once we recognize that chemical, it's going to be picking the chemical up and move to the right location, deposit the chemicals, and place the chemical back. So we actually can also achieve very, very high consistency of the droplet printing, and with less than 3% of CV, and have a high consistency of control of the droplet. We can also use the pressure and the nozzle size to control different droplet size, and so we can achieve a different volume control range for different applications. So here are some very simple patterns we deposited with the robotic hand, and you can see now we can fully utilize the precision and automation from the robots. Instead of doing our customized microfluidic fabrication, we can generate a standard nozzle head, but deposit all kinds of patterns and volumes we desire to have. So here is a video. You can see this microfluidic box is using the eyes in the front to recognize what the chemicals they like to pick up. And there's a processing unit on the back to recognize. And here is the fingers. The finger is actually grabbing the desired system to print. So once they recognize the right chemicals, they were going to use a grabber to grab the microfluidic system mechanically and move to the desired location. And there are two pneumatic control. One is to push the liquid to the nozzle head, and you can see here is the nozzle head. And on the back, also there's a pulse flow, and once you turn on the pulse, it will generate a droplet. So we can actually faster generate a minute droplets in a short period of time. And once you finish it with one, you can grab the second one and print the second chemical. So the robots can do a very simple job to complete a task that usually has to be done in a lab. Each of the droplets we printed here is about 100 nanoliter. So it's actually fairly precise deposition systems. You can actually see from the demonstration. And in order to showing the real application, we actually did a combinatorial array to show there's a protein expression experiment using bacterial array and lysis buffer. So from this picture, you can see we deposited an array with two combinations. One is a combination of the cell, a different concentration of the cell. And the two is a different concentration of the inducer, the chemical inducer we actually introduced to the system. Combine these two concentrations, we create a chemical gradient in 2 dimensions. So in this dimension, so we can see the cell and the plasmid concentration change. In this direction, we can see the inducer chemical change. So each of them come with different colors. And later we can observe the protein expression pattern. So as expected, with a higher cell concentration, with a higher the inducer level, we can actually see a higher fluorescent level from the pictures as well. So this work actually has been done with a collaborative expert with us and our collaborator in Chinese Academy of Science. And Jingjing was one of the lead science leader on the project. Kai is the robotic expert. Chuqing is doing a lot of chemical work and biological reaction. So Sony is designing all the microfluidic network, and Connery is helping with the final experiment we have. And we have collaborated with many labs in UC Davis and throughout the world with CAS. And we'd like to acknowledge our funding agency for the support. And the reading part of the robots actually is reading a barcode. So if you scan the barcode, that will link you to the paper we have published on this topic. And thank you for your time, and we're looking for any questions you have.
Hey, thank you very much, Ray, for your interesting talk. There are a couple of questions. The first question from Xiaodi Shu in Singapore. His question is, can this printing technique applicable on paper substrate?
Yeah, so basically this is a kind of like a modeling the inkjet printing. So it turns that things into a droplet. So it can be on any substrate. So we can print on glass, we can print on papers, we can do in micro teeter plate well. So I mean everything you have printed in the past, so you can think about this will be a desired substrate you can use.
Okay, very good. Another question I'd like to ask you is, as you are expert in the field of droplet microfluidics. This type of work is very interesting. In the field, certainly it is a very hot topic that people have been working on for the last decade. If you compare to the traditional wheel-based technology, we clearly see the advantages of droplet diagnostics. But what I would like to know, maybe you can share the insights from yourself, perspective from your side. What is the major challenge for this technology to be applied to clinical trial?
Well, I think that's a very nice question, and it's probably a broader question. I mean, I probably can talk because of the time limited. I probably only talk two aspects. I think current limitation. One is the whole system level. If we look at the conventional micro teeter plate based strategy, is they already meet a challenge of the volume. So with the conventional pipette system, anything below 1 microliter is not precise. So we cannot do anything smaller. So that definitely connects to the cost and the reagent consuming. So the microfluidic droplet technology probably can miniaturize the reaction and make cost better. But the conventional microfluidic chip method is quite hard to develop because every time you have to make a new chip, you have to characterize them, you have to make it robust and consistent. So the development time is too long. So that's why the recent industrial trend is to use the droplet technology. The hope of the droplet technology is you can use one technology to do different assays. Okay. So BioRed, this company actually has been a pioneer on this. It's actually been quite successful to introduce droplet PCR to the market, to one basically platform to have an assay and a highly precise manner can generate 2,000 droplets, which actually can drastically increase the dynamic range of the reaction. So which has been considered as two is for the whole system level, the system we are creating, one challenge versus the conventional microfluidic. Also, to react a the system. Okay, so that's the advantage of microfluidic chip. So everything is contained inside to react. The open system, like a plate, and also the printing system we have is open system. So there is a possibility of aerosol. Okay, so we're actually working on a combined system today to combine the droplet system with the microfluidic chip system, but we want to standardize the microfluidic chip system. So eventually, our hope is we're able to provide the laboratory with different sort with a standard microfluidic chip, one type, and then they can use a robot to do the customization. So instead of doing all fabrication of difficult microfluidic chips, they can program the robots. So which is much more pleasant job for many graduate students and the researchers as well.
Okay, thank you very much, Ting Rui, for your insights. Actually, I will receive. Due to the time limitation, let's thanks Professor Pan for the wonderful talk. Thank you very much.
Thank you.
Ting Rui.
Thank you, Tianhong. Thank you all.
Thank you. All right, let me introduce the last speaker, Dr. Yang Wang from Chinese Academy of Sciences. So he got his PhD from Chinese Academy of Sciences with distinction. And actually, during his PhD candidacy and right after, he received the Exceptional Achievement Award from Chinese Academy of Sciences. That's actually one of the major award for graduate students at Chinese Academy of Sciences. Only a few every year who can get that type of award. And then he worked in Peking University for a while. Now he is a research associate at Chinese Academy of Sciences. The topic he is going to talk today is microsensors for COVID-19, very hot topic. And actually his work currently is under clinical trial in Huaxi Hospital, which is one of the best hospital in China, just like a Mayo Clinic in the United States. So with this, Yang, please take your time to make your presentation.
Yeah, thank you. Thanks for Professor's introduction. And I will start. Good morning, everyone. I'm Yang Wang from Institute of Electronics, Chinese Academy of Sciences. It's my honor to have this opportunity to share my recently work with you, which is a laser-engraved, low-cost microvial platform for point-of-care diagnosis of COVID-19. And as we all know, this year on July 30th, the World Health Organization declared a global public health emergency over the outbreak of a novel coronavirus. And then on February 11th, the WHO officially named the disease as the COVID-19. And we know that the virus consists of 4 structural proteins and a single-stranded RNA as the genetic material. Up to now, the disease has spread to at least 200 countries and infected over 8 million people and resulted in at least 430. Well, as we all know, this year on July 30th, the World Health Organization declared a global public health emergency over the outbreak of a novel coronavirus. And then on February 11th, the WHO officially named the disease as COVID-19. And we know the virus consists of 4 structural proteins and a single-stranded RNA as a genetic material. And up to now, the virus has spread to at least 200 countries and infected over 8 million people and resulted in at least 430,000 deaths. What's more, the symptoms of the patients with the infection of COVID-19 are non-specific, and up to now, no vaccines or drugs have globally recognized and reported to be specific for the COVID-19. So to slow down the spread of COVID-19, lots of people have been placed under lockdown, which brings a big impact on the economic development. So to inhibit it from further spreading, there is an urgent need on the techniques for rapid yet accurate detection of COVID-19 in real time while offering low cost, easy operation, and highly sensitive. And then from this figure, we can see that various diagnosis methods have been developed for COVID-19 so far. And at present, the most commonly used method for the detection of COVID-19 can be divided into 2 primary approaches, including an antibody-based method and a nucleic acid-based method. And in general, the antibodies are mostly detected based on blood cells, and while the nucleic acid-based methods are commonly detected based on throat swabs, and they are used to directly detect the viruses. And as to the antibody-based methods, the basic mechanism is to detect the antibodies, including the IgG and IgM. And we can see from this figure that these two antibodies appear in human body successively after once you are infected by the virus. And through effective treatment, the IgM will disappear gradually and the IgG will persisted. And as reported, the antibody-based method targeting at IgG or IgM are consistent with the results obtained by nucleic acid-based methods. And nowadays, the ELISA and the lateral flow assay are commonly used for antibody-based detections. And now, generally, a clinical diagnosis via antibody methods can be finalized in as little as 5 to 15 minutes. And so together with the advantages of relatively cheap and easy to operate, the antibody-based methods are more suitable for large-scale preliminary screening of the COVID-19. Well, as to the nucleic acid-based method, with advances in molecular biology technologies, these methods have been developed rapidly and become a revolutionary technology for virus detection, especially for the COVID-19 detection. And in the nucleic acid methods, the method based on PCR is characterized by rapid detection, high sensitive and high specificity, which has been regarded as the gold standard for the COVID-19 detection. However, and we can see from this, the PCR detection usually takes 4 steps, including sample collection, sometimes sample transportation, and sample preparation, real-time PCR, and the last step is analysis and report, indicating that PCR usually needs a relatively long time. And what's more, PCR detections typically rely on expensive thermocycler and readout systems. These all greatly limited the size of the PCR's applications. And by contrast, several isothermal application techniques, which are conducted at a single temperature, and they do not need specialized thermocycling instruments. So as a result, they are ideal candidates for the point of care testing, ranging from community hospital to bedside. And we can see from this table that among these isothermal methods, including RPA, NASPA, HDA, and the LAMP, we can see that the LAMP only needs 1 enzyme and is suitable for various detection methods, including GERT and so on. And so it has emerged as a low-cost alternative for the rapid detection of COVID-19. And the LAMP reaction usually relies on 4 to 6 specifically, specially designed primers. And by using these primers, the target nucleic acid fragments of the virus can be identified and amplified at a constant temperature, usually from 60 degrees to 65 degrees within 1 hour or less. And what's more, without loss of sensitivity or specificity. And the LAMP usually could be used for detection of DNAs. As to the RNA virus, we only need an additional reverse transcriptors to convert the RNA into DNA, and then we can detect the RNA virus by LAMP. And what's more, after application, the results of LAMP could be usually detected in several ways, including by adding a coupled pH indicator or a fluorescent dye in the reaction buffer, or just using a lateral flow assay, and so on. So based on the LAMP technique, we proposed and fabricated a microware platform which can be used for multiplexed and point of care.
Can you see it now?
Yes.
Yeah, this is an announcement for those who are interested in microsensors, microfluidics. We have a 3-day conference, and actually the last day is for award. The so-called MEN 2020. This conference started from 2014, sponsored by a journal, Nature journal, a Microsystem and Nanoengineering. So this year, because of the coronavirus, so we move the conference online for Young Scientists Forum. The conference is on July 7 to July 9th. The 10th is for award ceremony. So the time is actually in Eastern time is 9 to 12 every day. So this is actually the conference. We currently have 54 young scientists globally, including those who from University of California System and MIT, Chinese Academy of Sciences. They are going to make their presentations on these 3 days. This conference was sponsored by the journal, Nature journal, so-called Microsystem and Nanoengineering. A lot of work on microsensors, microfluidics, microactuators, drug delivery systems, and those who are interested in this type of work, you can look into this journal and read the journal online. And actually, all the papers are open access. You can easily to download any paper that you are interested in. All you need to do is just get to the website on the footnote, www.nature.com/micronano. This is actually a journal sponsored by Nature Publishing Group in UK and Chinese Academy of Sciences. As far as we know, this is the first engineer journal initialized by Nature Publishing Group. So you can get all the works relevant to microsystem, microfluidics, sensors, and actuators, and so on and so forth. So with this, I'd like to stop my share. I hope that Dr. Wang can go back to his talk. So Yang, are you available to bring back to the conference?
I will start from this page.
Very good.
As I mentioned earlier, based on the LAMP reaction, we proposed and fabricated a microvalve array platform, which can be used for multiplex and point-of-care detection of COVID-19. And the design and the working principle of our proposed novel device is shown in the figure. As we can see, the extracted nucleic acid fragments are transferred into the microvalves where the LAMP reaction buffers has already been added, including primers, enzymes, and so on. So once the samples were added in the presence of the target RNA, a reverse transcription and the LAMP application process will be initiated. And we can say that our device integrates the functions of nucleic application and visual detection. So for the purpose of rapid and easy readout, we applied a colorimetric pH indicator in the system. And as we can see from this figure E, after application, the color of the mix will change from pink to yellow, and which can be read directly by the naked eyes. So we can modify our device according to different situations, and the device can enable multiplex detection from several up to hundreds of samples in one run. For quick proof of concept in this work, we fabricated a chip like this with 5 microvalves, which can be used for detection of 5 targets simultaneously. So to minimize the risk of false positive and false negative results, we also added 2 internal controls in the device. The negative control by using the BRCA1 gene, by the positive control by using the BRCA1 gene, and the negative control by using double diluted water in the device. And by adding this dual control design, which will not only help to improve the detection accuracy, but also help normalize the signal intensity, also enabling quantitative analysis. And what's more, the microvalve in the device was of special design, and we can see from this figure, we adopted a conical shaped microvalve, and this shape can not only to prevent the reaction buffer from evaporation, but also can help improve the heat transfer efficiency, resulting in a faster testing speed and detection accuracy. And the device is fabricated by using laser, and which is quite low cost. So the device we fabricated enables the advantages of rapid detection and low cost and high throughput detection, and easy to read by naked eye, and internal controls were involved in the device. And like I mentioned before, we use the lamp as our detection methods. So we designed 6 specific primers, including 2 inner primers, 2 outer primers, and 2 loop primers, target at the ORF1A and B gene. And this gene is said to be with high specificity. I'm sorry here about this because our paper and the patents are under review, so the detailed sequences of the primers are highlighted here. And from this figure, we can see that the real-time application results of the primers. We can see that all 3 sets of the primers we designed can be used for the application and detection. And the application properties seems almost the same at higher concentration of targets. However, when it comes to the lower concentrations, compared with the other 2 sets of primers, the time needed for the application based on the first set of primer is much less, which means that this set of primer has a much faster reaction speed. And in addition, we optimized different reaction temperatures ranging from 50 degree to 68 degree for the lamp reaction. And we can see from the results of agarose gels, the application reaches its best performance when the temperature was about 60 degree and 63 degree respectively. So we adopted 60 degree as a temperature for the reaction in the next works. And under optimal conditions, the sensitivity of the assay was evaluated both in solution and on the multiplexed microvalve biochip. And different concentrations of ORF1A and B gene plasmids were prepared. And we can see from figure A, the real-time application results show that the limit of detection of the virus in solution is about 1,000 copies per milliliter. And also we can see from figure D that the multiplexed microvalve also enables a similar sensitivity, and the yellow is a positive signal, and the pink is the negative signal. And also from figure C, we can see that our device could reach a faster detection speed with about 20 minutes. And to validate the proposed multiplex device, a double-blind clinical trial was conducted with 113 fully characterized clinical samples collected from West China Hospital. And West China Hospital is a very famous hospital, and it consistently ranked in the top 3 in China. And these samples has already been tested with the RT-qPCR primary to our trials. And we can see from this figure that 87 individuals have been confirmed with the infection, while 26 were negative for the virus. And we sent our micro devices to the hospital, and then they detected the samples independently. And based on the comparison of with the standard benchmark RT-qPCR, and we can see that our device only fails to detect 4 weakly positive samples and 2 negative samples, inhibiting both high sensitivity and high specificity of 95.4% and 92.3% respectively. And what's more, to cooperate with the microbial array platform, we are now developing an instrument which integrates functions of nucleic acid extractions and the rapid application and detection. And we assume that the instrument will be suitable for the following 4 application scenarios, including at home, at hospital, at train station and airport. And also the instrument enables functions of high throughput rapid detection within 25 minutes, high sensitivity can reach out to 1,000 copies per milliliter, and easy readout by naked eye. And that's all of my presentation. Thank you.
I'm muted. Thank you, Dr. Wang, for your nice, presentation. There are 2 or 3 questions, short one. The first question is how to shorten the response time of your assay. What's the next step?
Yeah, as to the time consumption, we meant to develop another device which uses the method of RPA. RPA is a much faster isothermal application method, which usually uses about 10 minutes for the application and the detection. And then we are now do research about this.
Okay, very good. This is a question from Wenzheng He from Tsinghua University. The second question from Yilin Liu. What is the mechanism of color change in the nucleic acid detection readout?
Well, as I mentioned before, we added a pH indicator in the reaction buffer, and with the target, the lamp reaction will initiate, and the pH of the reaction buffer will change. So the pH indicator will with this change, it will change from pink to yellow. And so it means when it's negative, the color will remain pink. And when it's negative, positive, the color will change from pink to yellow. Yeah.
Okay, next question, quick one. Xiaodi Shu from Singapore. How is the 1,000 copies per milliliter detection limits comparable to the RT-PCR? And is the clinical sensitivity related to the limit of detection of the test?
Yeah, as to this question, I have to say that the 1,000 copies per milliliter the sensitivity is lower than the RT-qPCR now. And so in the clinic, we can see from this slides, and so we fail to detect 4 positive samples. I think it may because of the reason that the limit of detection fails to detect the sample.
Okay, maybe you may take a last question. This is a question from Jitong Duan at University of Minnesota. What's the major difference between your technology and the current existing ones? As you know, they also have other colorimetric approach that people have been using for coronavirus detection. What's the major difference?
Yeah, I have to say that the major difference is between my work and the current testing method is that we fabricated a multiplexed microbial array chip, and this chip could and we included 2 internal controls in this chip, and which will minimize the false positive and the false negative results. And we can see that this chip was fabricated by laser, so we can easily change the device to meet the demands of different situations, and we can detect 3 to 64, including even up to 100 samples in one run.
And then this is probably a very short last question. What is the substrate material for your wells?
Well,
polymer.
Polymer. PMMA. The sensor was made of PMMA, one kind of polymer.
Okay. With this, I would like to thank you for your wonderful talk. I would like to thank Alex, Tingri, Yang for the 3 invited talks. Also, thanks all the audience to attend this section. Thank you very much. Let's adjourn this meeting. There will be another one next Tuesday. Thank you very much. Have a good day. Bye.
Yeah, thank you, Professor.
Okay, bye-bye.